There is a village in Ethiopia, in the highland plateau east of the Rift Valley, where two men might live a few hundred meters apart, speak the same language, eat the same food, and bury their dead in the same stony soil. In every observable way, they are neighbors. They are, in the common usage of the word, the same kind of person. Their DNA tells a different story. Those two men could be more genetically different from each other than either of them is from a Norwegian farmer, whose ancestors have lived above the Arctic Circle for thousands of years. The genetic distance between neighbors in the same Ethiopian village can exceed the genetic distance between continents, between Africa and Europe, because Africa is where the human story began, and where it ran longest before any of us left. That finding overturned something most people carry as an assumption so basic it never gets examined. The idea that visible differences, skin tone, eye shape, the architecture of a face represent deep biological divisions between human groups. The idea that the people who look most different are most different, all the way down. The data disagrees, fundamentally, repeatably, across every population scientists have ever sampled. This is what the largest genetic survey of human diversity ever assembled actually found. The Human Genome Diversity Project took shape in the early 1990s. The goal was to collect genetic material from populations across the full geographic spread of human settlement. Indigenous communities in the Amazon, nomadic groups on the Mongolian steppe, isolated island populations in the Pacific, remote Himalayan villages, dozens of communities that had remained relatively insulated from the large-scale movement of people that globalization was beginning to accelerate. The project was controversial from the start. Critics raised genuine ethical concerns about research on communities that had little history of benefiting from the institutions studying them. Supporters argued the data was irreplaceable. What no one disputed was that the data, once collected, would be the most comprehensive picture of human genetic variation ever assembled. The project ultimately sampled over 1,000 individuals from 52 populations across 7 geographic regions. Each sample was processed through sequencing technology that could read millions of genetic markers in a single run. The raw output filled entire server farms. When the first analyses came back, researchers noticed something immediately. The genetic differences between what we commonly call racial groups were smaller, much smaller, than almost anyone had anticipated. A person from Beijing and a person from Stockholm look substantially different by every visible measure: skin tone, hair texture, facial geometry. At the level of DNA, they share approximately 99.9% of their genetic code. That shared fraction is not a rounding error. It is the entire weight of our common evolutionary history, accumulated over hundreds of thousands of years before any of these surface differences existed. The remaining 0.1% was where things became genuinely complicated, because what that variation showed was not what the conventional racial framework predicted. Traditional racial categories, the typologies that European scientists constructed in the 18th and 19th centuries, and that subsequently shaped medicine, law, policy, and popular imagination for 2 centuries, had almost no coherent basis in the actual genetic data. The boundaries researchers expected to find between racial groups simply did not appear where the categories said they should. Someone from East Africa might share more genetic markers with someone from Southern Europe than with someone from West Africa. A Native American community in the Amazon carried unexpected genetic signals found in populations from Papua New Guinea. The categories that generations of scientists, administrators, and ordinary people had treated as obvious and natural turned out to be lines drawn over a continuous gradient boundaries imposed on data that contained no such divisions. But the deepest finding in the dataset was not about the similarities between distant groups. It was about diversity within Africa. Modern humans originated in Africa, current evidence indicates, somewhere around 300,000 years ago. For the overwhelming majority of that time, roughly 230,000 years, our ancestors remained on the African continent. Populations diverged, mixed, adapted to radically different environments across a continent of over 30 million square kilometers, and accumulated genetic variation across hundreds of thousands of years. The genetic diversity that resulted from that long, complex African prehistory is the foundation of everything that came after. When small groups finally migrated out of Africa, the current best estimate places this at roughly 70,000 years ago. Though the picture continues to be refined, they carried only a fraction of that diversity with them. This is the founder effect. A pioneering population is always a sample of the population it left behind, never the full set. The smaller the founding group, the narrower the genetic slice they represent. Think of it this way. Imagine a bag containing 100 marbles in every color, 100 distinct genetic variants representing African diversity. A small group picks up 15 marbles at random and carries them out of the continent. Those 15 marbles become the genetic foundation of every human population in Europe, Asia, the Americas, and the Pacific. No matter how those 15 marbles multiply and diverge across thousands of generations, they will never produce more variety than was in the original bag. They started as a subset. They remain a subset. The data confirmed this pattern across every migration route researchers traced. Populations became progressively less genetically diverse the further their ancestors had traveled from Africa. European populations showed less variation than Middle Eastern populations. East Asian populations showed less than Central Asian ones. Native American populations whose ancestors crossed the Bering Land Bridge in one of the last major pre-contact migrations, having traveled the furthest of any group from the original source, showed the narrowest genetic diversity of all. Distance from Africa, it turned out, is one of the single strongest predictors of a population's genetic diversity. Not because people who left were better or worse, stronger or weaker, because departure itself was a filter, and the further you traveled, the more filters you passed through. Now consider what this means for the biological basis of race. The traits we use to assign racial categories, the traits that have been used to justify enslavement, colonialism, and genocide for centuries are primarily physical: skin color, hair texture, the fold of an eyelid. These traits are encoded by less than 0.1% of the genome. They are among the most recent adaptations in human evolutionary history. Light skin in European populations emerged as an adaptation to lower ultraviolet northern latitudes, and it appeared only around 8,000 years ago. Blue eyes are even younger, tracing to a single ancestral mutation that arose somewhere near the Black Sea, roughly 6,000 to 10,000 years ago. The epicanthic fold characteristic of many East Asian populations evolved through an entirely separate genetic mechanism, likely as protection against cold wind and reflective snow glare. These features developed independently, through different genetic pathways, in response to different environmental pressures. They are adaptations, recent, surface-level, environmentally driven adjustments to local conditions. The traits that carry far more genetic complexity, immune function, metabolic processing, disease resistance, neurological development show massive variation within every population group. You cannot reliably predict any of these from appearance. The genes relevant to them are distributed across the entire human species without respecting any racial boundary. The same principle applies to the geographic clustering of certain genetic diseases. Sickle cell anemia appears at higher frequencies in people with ancestry from malaria endemic regions West Africa, but also parts of the Mediterranean and South Asia. The sickle cell variant protects against malaria when inherited in a single copy. Natural selection favored it in regions where malaria killed regularly, regardless of what racial category the affected populations would later be assigned. A West African and a historically malarious region Southern European might both carry that variant. Two people classified as the same race, could have completely different susceptibility profiles depending on where their actual ancestors lived and what diseases those ancestors faced. These patterns are records of local evolutionary responses to specific threats. They are not evidence of fundamental divisions between racial groups. A doctor who treats racial category as a reliable proxy for genetic disease risk is substituting a social construct for actual information, and the actual information, where available, is far more precise. So where did the concept of biological race come from, if not from biology? European scientists in the 18th and 19th centuries attempted to classify human variation using the same taxonomic methods they applied to plants and animals. They built hierarchies from physical appearance. They ranked populations by supposed developmental level. They used those rankings to provide intellectual scaffolding for systems of colonial extraction and chattel slavery that were already in operation. The science followed the politics. The racial categories served the needs of the people constructing them. Modern genomics dismantled the biological basis for those hierarchies. The traits early race scientists used to define and rank populations have no measurable correlation with intelligence, social complexity, or any other criterion of human worth. The genetic differences that do exist between populations are either neutral or adaptive responses to local environments, not markers of a developmental hierarchy. But perhaps the finding with the most direct bearing on how we understand our own ancestry, our own family trees, is the one about universal common descent. Based on population genetic modeling, the average time back to the most recent common ancestor of all living humans is estimated at roughly 3,000 years. That is approximately 120 generations. Go back 120 generations, and you will find an individual who appears in the family tree of every single person alive on Earth today. Not metaphorically, mathematically, the family trees of a person from Lagos, and a person from Oslo, and a person from Osaka converge within recorded history. Going back further to roughly 7,000 years ago, every person alive today who has any surviving lineage is an ancestor of every person alive today. Every farmer in ancient Mesopotamia whose descendants made it to the present is your ancestor. They are also the ancestor of every person reading this sentence from any country, any community, any background. The idea of a pure ancestral lineage, a straight, uncontaminated genetic line from a single ancient origin point, is not just scientifically unsupported. It is mathematically incoherent. Your ancestry is not a line. It is a web. It encompasses the entire human family tree, because after sufficient generations, that is the only web there is. The same complexity appears in the histories of populations we often think of as coherent, unified groups. Modern Europeans, for example, are the genetic product of at least 3 distinct ancestral populations. The hunter-gatherers who first colonized the continent after the last glacial maximum. Anatolian farmers who arrived roughly 9,000 years ago and largely replaced those original inhabitants, and Yamnaya, pastoralists from the Pontic-Caspian steppe, who arrived around 5,000 years ago and reshaped the genetic landscape a second time. None of these groups had a concept of being European. None of them were genetically pure. They were already mixtures of earlier movements. The Han Chinese population, often treated as the paradigmatic example of a racially and culturally unified group, actually represents the convergence of distinct northern and southern populations that merged over thousands of years of expansion and state building. The genetic diversity within China's present-day population rivals the diversity across the entirety of Europe. Everywhere the data set pointed, it found the same thing. Human genetic variation is continuous, not categorical. Populations blend into each other across gradients without sharp dividing lines. The differences within any named racial group are consistently larger than the average differences between groups. The categories we invented to divide humanity do not describe anything real in the genome. Race is a social technology, not a biological fact. It was built to serve specific historical purposes, and it has served them with devastating efficiency for centuries. But it was not discovered in nature, it was constructed, and the genetic record shows, with increasing clarity and resolution each time we look, that the construction was always fiction. This does not dissolve the consequences. The lived experience of people categorized into different racial groups has been shaped by those categories in ways that are measurable, material, and ongoing. The disparities in health outcomes, wealth, educational access, and exposure to state violence that exist between racialized groups are not genetic in origin. They are the accumulated products of social systems that treated a fiction as biology and enforced that fiction with laws, violence, and institutional power for generations. The genomic data does not erase that history, but it removes the last intellectual cover from those who would blame the outcomes on the people who suffered them. What the scientists found, after decades of sample collection and analysis, after building the largest human diversity database ever assembled, was not a hierarchy. It was not a set of natural categories. It was a single, recently diverged species, carrying within it the accumulated variation of hundreds of thousands of years of African prehistory, sorted by local adaptation into the visible spectrum of human physical diversity, a spectrum with no meaningful breaks in it, no dividing lines, no genetic borders. The differences that drove centuries of catastrophe were never as deep as the people inflicting that catastrophe needed them to be. The bones knew, the DNA confirmed it. What the research also found in the margins of every dataset was evidence of how much the story is still being written. Some Native American populations carry genetic signals related to populations from Oceania, Aboriginal Australians, Papua New Guineans whose presence in ancient American genomes remains one of the field's open puzzles. The mechanisms of early human migration were more complex, more global, and stranger than any linear model of continental spread has yet captured. The data answered certain questions definitively. It opened others that researchers are still working to understand. That, perhaps, is the most honest summary of where the science stands. What we are is clearer than it has ever been. How we got here is still being discovered.